Good morning, everyone. Welcome to today's presentation. I'm excited to share with you some fascinating insights into life cycles in algae. My name is Dr. A.M. Anandakumar, and I am an assistant professor in the PG and Research Department of Botany at NGM College, Palachi. Today, we will explore the various types of life cycles that algae undergo, understanding their complexity and significance. So, let's dive in and discover the intriguing world of algae together. Let's take a closer look at the haplontic life cycle. This is considered the simplest and most primitive type of life cycle in algae. Interestingly, many other types of life cycles actually originate from this one. You'll find this pattern in almost all chlorophyceae, with just a few exceptions. Sometimes, this cycle is referred to as the Ulithrix or Chlamydomonad type, which helps us connect it to specific examples. In this cycle, the somatic or vegetative phase, which is the main body of the plant, is haploid, meaning it has just one set of chromosomes. This haploid plant is called the gametophyte. The diploid phase is very brief and is represented by the zygote, which forms when two haploid gametes fuse. During germination, this zygote, which is diploid, undergoes meiosis. This process produces haploid zoospores, which are essentially spores capable of developing into new plants. These zoospores or aplanospores develop into individual plants, and this cycle continues. In the haplontic cycle, the plants themselves, like Chlamydomonas, Ulithrix, Edygonium, and Spirogyra, are haploid. The main vegetative body is haploid, and the diploid phase appears only briefly as the zygote. So, in summary, the key points are that the haploid phase is dominant, the diploid phase is limited to the zygote, and this cycle is fundamental to understanding how many algae reproduce and grow. In this slide, we're looking at the diplontic life cycle, which is actually the reverse of the haplontic type. So, in the diplontic cycle, the somatic or body phase of the plant is diploid, meaning it has two sets of chromosomes. We refer to this as the sporophyte stage, which is 2N. The haploid phase, which involves the formation of gametes, is quite restricted and only occurs during gamete formation itself. Now, these gametes are haploid, or N, and they're produced by meiosis from the diploid sporophyte. When two gametes fuse during fertilization, they form a diploid zygote. This zygote then develops into a new diploid sporophyte through mitotic divisions, continuing the cycle. It's important to note that in plants exhibiting this cycle, the plant body is diploid, and the sporophytic sex organs are also diploid. The process of meiosis is crucial here because it forms the haploid gametes from the diploid sporophyte. The haploid stage is only represented during the formation of these gametes. As an example, groups like Bacillariophyceae, Sargassum, Fucus, and other Phaeophyceae follow this diplontic pattern. Overall, this cycle highlights how the plant's dominant phase is diploid, with the haploid phase being temporary and restricted to gamete formation, which is quite a contrast to other life cycles we've studied. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at the isomorphic alternation of generations. In this type of life cycle, you find two somatic phases that are exactly similar in appearance and structure. That's what we mean by morphologically identical. These phases are both plant forms, but they differ in their genetic makeup. One is diploid, called the sporophyte, which has two sets of chromosomes, and the other is haploid, known as the gametophyte, with just one set. What's interesting here is that the two phases look alike, which makes this alternation quite distinct from other types where the generations are visibly different. Among the green algae, specifically the chlorophyceae, this pattern is observed in groups like Ulvaceae, Chaetophoraceae, and Cladoforaceae. You also see this kind of alternation in certain brown algae, particularly in the orders Ectocarpales, Cuttlerails, Tylepteridales, Sphacelarails, and Dictyodales of the Phaeophyceae class. Now let's walk through the cycle itself. The zygote develops into a diploid multicellular plant, which we call the sporophyte, and it does so by postponing meiosis initially. Before the formation of spores, meiosis actually occurs, producing haploid spores called zoospores or meiospores. These spores then develop into haploid plants, the gametophytes. Finally, the gametophytes produce gametes, which fuse to form a new zygote, completing the cycle. So, in essence, there's a beautiful balance here. Both phases look similar, but they carry different genetic information, and this alternation is fundamental in understanding the complexity of plant life cycles. Let's talk about the diplohaplontic life cycle now. In this cycle, both 1N and 2N plants are present, which means we see both haploid and diploid stages. This type of life cycle actually has two main variations. The first is called isomorphic alternation of generations. Here, both the haploid and diploid forms look very similar morphologically. They alternate in the life cycle, but visually they are almost indistinguishable. Examples of this include algae like cladophora, ectocarpus, and ulva. The second type is heteromorphic alternation of generations. In this case, the two stages are quite different in appearance. The 2N plant is usually larger and macroscopic, meaning you can see it with the naked eye, while the 1N stage is microscopic. Laminaria is a good example of this type. Understanding these two types helps us grasp how complex and diverse plant life cycles can be, especially in algae, and how they adapt their reproductive strategies to their environment. This concept ties into the overall theme of plant evolution and diversity, showing how different organisms have evolved distinct ways to complete their life cycles. Let's talk about the diplobiontic type of life cycle. This type is actually found in almost all rhodophyceae, which are red algae, except for the nemmelionals. So it's quite widespread, especially in groups like Polysiphonia and the order Ceramiales. Now, the key thing to remember here is that the life cycle is triphasic, meaning it has three distinct phases. It involves an alternation between two diploid, or 2N, sporophytic generations. These are called the carposporophyte and the tetrasporophyte. In addition to these diploid phases, there's a single haploid, or N, gametophytic generation. So overall, we see two diploid phases and one haploid phase throughout the cycle. The gametophytes, which are the haploid phase, produce gametes. When these gametes unite, they form a zygote that is diploid, or 2N. This zygote then divides repeatedly, giving rise to the sporophyte generations. This cycle is fascinating because it highlights the complex but well-coordinated alternation of generations, which is a key theme in understanding the life history of red algae. It's a perfect example of how different phases work together to ensure the survival and reproduction of the organism. In this slide, we are exploring the haplobiontic life cycle, also known as the diphasic life cycle, which features three distinct phases in the life cycle. Out of these three phases, two are haploid, meaning they have N chromosomes, and one is diploid, with two N chromosomes. Examples of organisms exhibiting this pattern are found among nemmelionals, like Batrachospermum, which belongs to the rhodophyceae, and Collochaete from the chlorophyceae. Now, focusing on Batrachospermum, the haploid gametophytic phase produces gametes, and when these gametes fuse, they form a zygote that is diploid, or 2N. This zygote is the only diploid phase in this cycle. It then divides meiotically, which leads to the formation of a haploid, asexual phase called the carposporophyte. The carposporophyte reproduces asexually by producing haploid spores called carpospores. These spores develop into new gametophytes, which are haploid plants with N chromosomes. So, essentially, we see an alternation between two morphologically different haploid phases, the gametophyte and the carposporophyte, with the zygote acting as the diploid stage bridging them. It's important to note that in this cycle, there are two well-developed haploid phases, the gametophyte and the carposporophyte, and only a single diploid phase, which is represented by the zygote. The haploid generation includes the independent gametophyte, its sex organs, and the gametes, while the second haploid phase involves structures like gonomoblast filaments, carposporangia, carpospores, and the chantrantia stage. We call this a diphasic life cycle because both haploid phases are distinctly present and functional. This type of life cycle, called haplobiontic, is characteristic of organisms like Batrachospermum and Mammalian. Overall, this cycle exemplifies how complex life cycles can be, with multiple haploid stages that alternate with a single diploid stage, highlighting the diversity and adaptability within algal reproductive strategies. Let's take a closer look at the diplobiontic life cycle, which is also known as a triphasic life cycle. This means it involves one well-developed haploid phase and two diploid phases. The key idea here is that the organism alternates between these different stages during its life. Starting with the haploid phase, this is represented by the male and female gametophytic plant bodies. These are the structures that produce gametes, sperm, and eggs, and are responsible for sexual reproduction. Think of these as the reproductive groundwork of the cycle. Next, we move to the first diploid phase, which is a bit more complex. It begins with the zygote, formed after fertilization, and then develops into structures like gonomoblest filaments, carposporangia, and carpospores. Collectively, these form what's called the carposporophyte. An important point here is that this phase depends on the gametophyte for its existence, meaning it remains attached and reliant on the haploid plant. Finally, we reach the second diploid phase, represented by the tetrasporophyte. This is a plant that bears tetrasporangia, which produce tetraspores through meiosis. These spores will eventually grow into new haploid gametophytes, completing the cycle. An example of this cycle in action can be seen in Polysophonia, which vividly demonstrates these stages. So, the overall theme here is the intricate dance between haploid and diploid stages, each playing a crucial role in the life cycle, ensuring the continuity and diversity of the species. This triphasic cycle is a fascinating example of plant reproductive strategies.